MIGAL, Galilee Research Institute, who will speak about the race to find a cure for the coronavirus. I want to thank my colleagues at the Jerusalem Press Club for facilitating this webinar. Mr. Ben Herzl will start with a short introduction and then we'll open up for Q&A. You are on mute, so please type your questions on chat and he will answer them. Ori Ben Herzl has been appointed CEO of MIGAL recently. Previously, he was Director of Technology of Kaima, BioAgritech. And a word about MIGAL, it's a Galilee Research Institute. It's an applied research organization specializing in the field of biotechnology, environmental and agricultural sciences. It was established in 1979 in the city of Kiryat Shmona to create a scientific research facility and knowledge-based economic generator at Israel's northern periphery. And for the past 4 years, the team of MIGAL scientists have been developing a vaccine against infectious bronchitis virus, which causes a disease affecting poultry. And we're hoping that there will be some similarity to the coronavirus that affect humans as well. And we'll hear more about it from Ori Ben Herzl. Mr. Ben Herzl, please.
So thank you very much, Uri, for this opportunity to speak here. One correction in your intro, I'm not the CEO, I'm a VP of Business Development. And I can start here our presentation where you can see some of the slides that talk about our technology. Just 1 second here. Okay, can you see that?
Yes, we can.
Okay. So I will start with just a few words background on MIGAL.
Ori, sorry. Guys, Ori will send us the presentation later. So if you miss something, rest assured that you'll get it later.
Yes. Just a few words on MIGAL. So MIGAL was established about 40 years ago in the upper north in Kiryat Shmona. It's as you know, it's the far periphery of Israel. It was established as a research institute with the clear vision to enhance the economic in the region and to attract researchers, to attract talent and industry to the region. As one of the founders, Professor Dan Levanon, that serves as the chief scientist of MIGAL, and also served many years as the chief scientist of Ministry of Agriculture, he told us when we started, when we just entered to this crisis, that MIGAL knew a lot of difficult times during the history since it was established. And one of most critical thing is to help the region and all the economy around the ecosystem to get through these difficult times, and even more important is to after it ends to help it develop and grow again after the crisis is over. So this is exactly what we are aiming to do here, actually by coincidence, and I will talk about it in a few slides later. So this is just a little bit the info about MIGAL. We are in a very tight relationship with Tel Chai College in the north. Our researchers are the teachers in Tel Chai. And we have about 300 employees and more than 70 PhDs that are working in 44 research groups. So it's kind of a big institute for research. We are working on research in almost everything between agriculture, food, feed, health, and environment. And we are set up in 4 departments in plant sciences, precision agriculture, and nutrition and health, and biotechnology. Biotechnology is actually the department that is more relevant for today. And this is where our research of development this vaccine started. So just a little bit background on vaccine development. So currently there are, very, I would say, traditional methods to develop a vaccine. There is a vaccine that is a weakened virus or a dead virus that is injected to the body, and then the immune system get to know the structure and the active domain of the virus, and then it will be more prepared for next time. So this development of this kind of vaccine is very intense. It includes a very wide screening of different types of mutants of viruses that are mutated by chemicals or some other agents to create damage to the virus, and then it gets tested. Most of the time, the researchers getting to that end, and really just few very low chances to get something through. This is why most of the vaccine development or traditional vaccine development is kind of take a lot of time. So today I'm going to present, I would say, much more innovative and modern way that we use to solve this problem. So the story starts maybe 4 years ago with the initiation of Ministry of Agriculture and their fund to create a knowledge center in viral diseases in poultry, chickens. They targeted 4 main viral diseases in this industry that create a lot of damage to the farmer, and they wanted to find a cure or find solution for this problem. So they set up this knowledge center that was established by interdisciplinary groups in our biotech department in MIGAL. So the team contains actually a virology and poultry vaccination expert, and this is the leader of the team, Professor Jacob Witkowski. Also immunologist, microbiologist, computational scientist, especially in chemistry, and protein and enzyme design by Itamar Yedid. We did it with collaboration with the Veterinary Institute, where we did the actual test for the chicken with live virus, and I will get to that. So this is the new or more modern way using computational tools to design a vaccine. So as you see here, this is the spike protein, and this is the outer surface protein that is very famous in this picture, and we actually could look on the how it looks in the on this actually structure, and not just the sequence, and the structure in the space is very important here. So you see IBV in the left, this is the virus for chickens that we worked on, and then you see some other types of spike protein of MERS and SARS, and then the COVID-19, this is the red one. So this is the way we can look on this protein, and decide whether the structure is similar enough to adjust our vaccine or not. By these tools, but by these computational tools that we use, our scientists actually looked at the sequence of the new COVID-19 in human when it was published. First, it was published, I think, maybe 3 months ago. And they compare it to the virus that they knew, which is the IBV, and I will get there quite soon. I think in the next slide. So our vaccine is a combination of two main subunits in the virus. One subunit is the S, the spike protein, which I mentioned last slide. Another subunit is the N, nucleocapsid, which is a part inside the virus that helps the virus to organize its RNA. So by targeting these two subunits, we can create a very good defense by teaching the immune system how to respond when it will find a real virus in the future. So what we are doing is we're actually synthesizing a chimeric protein, the S protein, the N protein, and we combine it with a carrier. This carrier is actually another a very important part in this development, and it's another kind of a protein that can deliver the subunits to the mucosal immune system. It's through the mouth or the nose or any other mucosal tissue of human beings or any other animal. So we use this carrier to actually deliver the protein to the target tissue, which is the upper respiratory system, and to create the immune response in the target tissue. So we have the carrier, we have the subunits, and we have the oral delivery or oral administration, which is delivering the subunits to the specific tissue in the organism. So as I mentioned, we started in poultry, and the 4 years we worked on poultry, and this is another very important thing here, that we had the time to learn how this virus responds to the immune system after we activating it. And what I show you here is actual results of or comparison between immune and non-immune chickens were infected by the live coronavirus.
You're coming across a bit broken.
COVID-19, and the time it took to clear the virus. Okay, sorry. Can you hear me now?
A bit better. Can I ask you to?
Can you hear me now?
Yeah,
Hello?
yeah.
Okay, let me see.
Yeah. I hope this is not too scientific to our participant.
Okay, no, you know, this is very important. It's not.
Yes.
I don't think it is actual living model where we show a challenge test model in chickens, where we challenge the immune or the vaccine that we created to the right column. You see the orange exposing the chicken to the virus, and then after 6 and 10 days, you see how fast the clearance of the virus was happened. And in comparison to other, just the S, just the N, some other control and so on, what we are trying to show here is that we actually did a live model with a coronavirus member, which is very difficult to do with the COVID-19.
Okay.
So the advantages of this technology or this vaccine, it cannot cause disease state because we are not using a weakened or dead virus. We are using the subunit. It's safe to use in immune suppressed patient because of the same reason I just explained. Less chances of side effects. This is because the oral delivery, oral administration, it makes it much easier to administrate with kids or with rural places where you don't have hospitals or healthcare. And we're creating a simulation of T cells. This is what actually the N subunit creates. And this is very important because it's holistic, more holistic immune response together with the S. And another very important thing here, because we designed the system for chickens, it has to be for the farmers, right? It has to be quite affordable. It has to be scalable. You need to produce a lot of doses in a relatively fast production. So we have a very efficient production method. And we can, as we just adjusted from avian corona to human corona, we will be able to do it even faster in the next variant, if there will be one. So I think that's it, and I will just say a few words about what our stage today, and then I can answer for some questions.
Okay.
Let me just see how I stop sharing. Okay. So so today we are in the middle of producing the new adjusted protein subunits for human. We are starting, and we are actually expect to done with preclinical trials in the next few weeks, and to be ready to start clinical trials in human. Now, we are in the middle of a process of learning the regulatory requirements in all authorities, Europe and the US. And we hope to, or we expect to apply the regulatory request in a few days. So basically, we are pretty sure that we have a vaccine that is quite safe. We have oral delivery. We are using subunit, which is actually protein. You can use it, or you can look at it as a food additive. And we have all the results that we done with chickens to prove safety. So with all this in mind, we are very confident that the safety phase will be quite fast for us. And then we will have to prove efficiency and so on. But it really depends on the authorities, and we still don't know exactly what the requirements will be and how much time it will take us to get through all the clinical trials. So I think this is most of it.
Okay.
Yeah.
Thank you, Ori. And again, we let everybody know that they can get the presentation. You'll send it. Can you also send some pictures of MIGAL so people can use them free of charge?
Okay, yeah.
There's a question, all the advantages you mentioned are for the poultry vaccine. And do you expect the same ones for the human, or it's impossible to know now?
So first of all, oral delivery is advantage. Also, I mean, this is very clear that it will be also advantage in human, and also the subunit, the protein, instead of weakened or dead virus is very significant. We are doing now these days additional trials in mice to prove the same results that we prove in poultry. Once we have it in mice, it will be very easy to claim that we can prove the same in human. And we expect to be in that place maybe, I don't know, very few weeks from now. So we expect the same advantages will continue when we'll be in human.
Okay, so another question is what brings your vaccine to be different from the other ones scientists are trying to develop? I'm sure you guys are following what's going on in other places.
Yes.
And why
Yes.
are you closer than anybody else?
So I don't know if we are closer or not. You know, this is what we are doing. What I do know is that there are about few tens of other companies and the research institute that are working to develop a vaccine. Most of them are working on or RNA, which is a different method, which haven't been commercially proved yet. And most of them are by injection, not orally delivery. And this is very important for this development. Another thing is the combination between the S subunit and the N subunit, which creates more holistic or more broad immune response, which is very important, especially to the respiratory system. So we are very confident in that.
There's an interview today with Professor Cohen, who's the president of Hebrew University, who says that he laments the fact that back then when the SARS plague was overcome, institutes basically stopped the research on it. So we lost a lot of years of research. Are you aware of this, and do you subscribe to this opinion?
No, so we know that they stopped the research when the SARS, also the MERS, just fade out. I'm not a scientist, but what we know is that there are some assumptions that this COVID-19 is here to stay, and we will need to start take this kind of vaccine or maybe another vaccine in through the years, like we do with influenza and some other viral diseases. But this is just an assumption. We don't know. Maybe it will just get to a very low infection rate and fade away from our life, which I really hope will happen, but we don't know.
It is also said or reported in the papers that the Biological Institute in Nestiona is pushing very intensely towards finding a vaccine, but they got the real virus from China, from others.
Yes.
This is an advantage, no?
Of course, this is an advantage. We do not use the live virus. So what I think, it's not just the Biological Institute in Nestiona, it's also other research institutes and the hospitals around the world they are now trying to develop the animal model to test the challenge test that I just showed you we done in chicken with the COVID-19 and not another variant from the same family. It will be very useful for all the vaccine and also other medicine development for corona. We don't see, I mean, we probably would like to use this method if they will have it. But other than that, I just know everything from the papers in that matter, and I don't. I mean, this is just my opinion. Yeah.
It seems like everybody is working in their own niche to find a solution. Isn't there an attempt to collaborate, share information, maybe to have a central thrust toward finding a cure?
So on the scientific level, I don't think this is true. I think on the scientific level, there is a lot of knowledge share, as we are sharing almost everything that we are doing with anybody that is showing interest. And we are very open to collaborate if it's for detection methods or curative aspects and so on. So we are very open, we are very collaborative, and also other institutes. I think since our first PR, we had maybe thousands of of companies or institutes, governments reaching out, and when it's relevant, we are continue to work together. But most of the times, if there is no mutual interest to continue, we just continue by ourselves or them. So it really depends on the same issue.
Last question. Tell us on a personal level, how do you work? How do you carry on the research there?
So we are working 24/7. The scientists, the research assistants are working all the time in the lab. We are not, we are trying to be very, very efficient, very fast. And we are very excited to be part of, the common effort to find some solution to this crisis.
Very good. There's a last question. I'm not sure you know how to answer it, but there's a question for Italian journalists. If you can say something about the situation in northern Italy.
No, I don't know much. I mean, just from the news, and that's it. I don't have any other insights than you guys.
Okay, Ori, thank you very much. The Lord said to Jeremiah, out of the north, the evil shall break forth upon all inhabitants of the land.
Yeah.
And we hope that out of the north, there will be a breakthrough in the search for a cure for the coronavirus. And if and when that happens, remember, you first heard about it here. Thank you very much for joining us. And heads up for tomorrow, 3 p.m., we have Shira Greenberg, who's the chief economist of the Ministry of Finance, who will speak about the new economic recovery plan. Thank you very much for joining us. Thank you, Ori, and good luck with your efforts.
Thank you very much.
Thank you.